In glycolysis, 1 mole of glucose is converted to pyruvate molecules in the cytosol of an animal cell. In this process, 2 NAD plus molecules forms 2 NADH molecules, and also 2 ATPs are formed by this process. These 2 pyruvates formed now pass through the membrane channel protein called porine present in the outer mitochondrial membrane. These porins make the outer mitochondrial membrane permeable to many metabolites. Then this pyruvate passes through the transporter protein present in the inner mitochondrial membrane and reaches to the mitochondrial matrix. In the mitochondrial matrix, the pyruvate is converted to acetyl coenzyme A. During this conversion, 2 NAD plus forms 2 NADH and a CO2 is formed during this conversion. The CO2 diffuses out from the matrix and reaches the intermembrane space. Now the acetyl coenzyme A undergo oxidation in Tca cycle and generates 3 NADH, 1 FADH2, 1 GTP and 2 CO2 molecules. The 2 CO2 molecules diffuse out from the matrix. Here the acetyl coenzyme A acts as connecting link between glycolysis and Tca cycle. The generation of GTP in Tca cycle is the characteristics of animal mitochondria. Now let us focus on Tca cycle. This cycle was given by Hans Krebs in 1937 and he got Nobel Prize for this work in 1953. During aerobic respiration in the cytosol of an animal cell, glycolysis occurs. In glycolysis, glucose forms 2 pyruvate molecules. Then one of the pyruvate molecules, in presence of a multiprotein complex called pyruvate dehydrogenase or PDH, forms acetyl coenzyme A. This is a oxidative decarboxylation reaction because the carbon atom of pyruvate is lost as CO2. In this reaction, the NAD plus forms a NADH. This reaction is also called as intermediate step in the aerobic respiration. The beta oxidation of fatty acids also produces acetyl coenzyme A. Now, in the first step of Krebs cycle, the acetyl group of acetyl coenzyme A condenses with oxaloacetate to form citrate. So, this cycle is also called as citric acid cycle. This citrate has 3 carboxylic acid groups. So, it is a tricarboxylic acid. For this reason, this pathway is also called as tricarboxylic acid cycle or Tca cycle. This condensation reaction is driven by free energy of hydrolysis of thioester bond. This step is also releases a H plus ion. This step is named as Tca1 and is catalyzed by citrate synthase. The H2O can hydrolyze the coenzyme A. This citrate is then converted to isocitrate. This step is named as Tca2 and is catalyzed by achotinase. The isocitrate is then converted to alpha ketoglutarate. This conversion releases a CO2 molecule and one NAD is reduced to NADH. This step is named as TCA3 and is catalyzed by isocitrate dehydrogenase. Then the alpha ketoglutarate forms succinyl coenzyme A with the release of a CO2 molecule. Here also NAD plus is reduced to NADH. This step is named as TCA4 and is catalyzed by alpha ketoglutarate dehydrogenase. The succinyl coenzyme A then forms succinate. This reaction also forms coenzyme A by the hydrolysis of thioester bond in succinyl coenzyme A. This coenzyme A can again combine with succinyl coenzyme A to form succinate. The hydrolysis of thioester bond in succinyl coenzyme A is coupled to a reaction in which the GTP and PI are combined to form GTP. This GTP then again can be converted to GDP with the release of PI. This released PI can combine with ATP to form ATP. So this step is called as ATP generating step. Here this step is named as TCA5 and is catalyzed by succinyl coenzyme A synthetase. Then the succinate forms the fumarate. In this reaction, the coenzyme FAD forms its reduced form called as FADH2. This step is named as TCA6 and is catalyzed by a membrane-attached enzyme called succinate dehydrogenase. The fumarate then combines with H2O to form malate. This step is named as TCA7 and is catalyzed by fumarate hydratase. Then the malate regenerates oxaloacetate with the conversion of NAD plus to NADH and a H plus ion. This step is named as TCA8 and is catalyzed by malate dehydrogenase. It is important to note that no molecular oxygen does not participate in Tca cycle. Now let us calculate the ATP gain in one complete cycle of aerobic respiration per 1 mole of glucose. First of all, we have to remember that one NADH2 goes through ETS or electron transport system in the mitochondrion and forms 3 ATPs. And one FADH2 goes through ETS to form 2 ATPs. In glycolysis, 2 NADH2 and 2 ATPs are formed. These 2 NADH2 molecules go through ETS to form 2 into 3 is equal to 6 ATP plus 2 ATP is equal to 8 ATP. In 2 intermediate steps, 2 NADH2 molecules are formed. These 2 NADH2 molecules go through ETS to form 2 into 3 is equal to 6 ATP. The 2 pyruvate molecules formed from glucose by glycolysis undergo two different Tca cycles. So two Tca cycles form 6 NADH2, 2 FADH2 and 2 ATP. These 6 NADH2 go through ETS to form 6 into 3 is equal to 18 ATP, and 2 FADH2 go through ETS to form 2 into 2 is equal to 4 ATP plus 2 ATP is equal to 24 ATP. So ATP gain in grand total, glycolysis forms 8 ATP, 2 intermediate steps form 6 ATP, 2 Tca cycles form 24 ATP. So total 38 ATP. Please like, subscribe and share.